Hoverfly Queen herself. Oh, sorry, I should have waited for that. Next, I'll just repeat that. Next up is Dr. Ellie Rotheray, who is our in-house hoverfly expert, and she's going to be talking about spreading the word using citizen science, Hoverfly Lagoons project case study. Over to you, Ellie.
Thanks, Dave. Yeah, I'm here to talk about the Hoverfly Lagoons project primarily, I'm just going to talk about the Hoverfly Lagoons project. I lead this project and I'm going to describe how this project came about, maybe more importantly why, and summarize some of the results and its success in spreading the word. So why the Hoverfly Lagoons project? Well, it's all Dave Goulston's fault, as he said at the beginning. He had this idea, which he's already introduced, and it was around 2015, I think, when we first kicked off, to create the citizen science organization, which would run these projects. And did anyone in his lab have an idea for it? And at the time, I was a postdoc in his lab. And I'd been pondering this idea for a little while already as a tool for maybe supporting and encouraging hoverflies in agricultural spaces, not so much in gardens. But this seemed like a good fit for what was to become the Buzz Club. And this is one of the first newsletters reporting on its results in that first year. For my PhD, I'd studied the ecology of the pine hoverfly in Scotland. And my studies focused on the larval stage, which is here in this image, in the central image. The adults, male and female, are at the top there, black with this rather lovely orange bottom. So hoverfly ecology is largely determined by their larval stage, by the larvae, their biology and feeding requirements, and they vary substantially within this group. Which makes hoverflies just generally as a group really fascinating group to study, and also really useful and important bioindicators. And the pine hoverfly within this group are semi-aquatic. They feed on microbes in water. They're saprophages, which means they depend on decaying organic matter. And I was making a range of habitats to try and mimic their known natural habitat, which was holes in tree stumps. I was just getting a pointer here. And this you can see here, this tree stump, there's a hole starting to emerge in the center of this stump, which is left over after the tree is being cut down. This is what would happen in a natural, more natural situation where the hollow of the stump of this nice old granny tree has snapped at the base, and inside here it would fill with rainwater and the larvae would develop in this rainwater. I wanted to see if simple containers filled with pine chips and sawdust would attract this species in the field, because we were limited by the number of pine stumps. We didn't want to cut down a load of trees. We wanted to boost the population where it was. It was super restricted and very few left in Scotland. And also to aid dispersal. We wanted to encourage them to disperse. So could we create stepping stones in the countryside using these artificial habitat containers filled with sawdust? I found that the larvae, oh, well, I was also rearing them in captivity. So I needed to recreate their habitat in a captive situation as well. And I found that the larvae actually can be reared on almost anything. They didn't seem to care about the kind of sawdust they were growing in. As long as they were microbes, they didn't seem to care. They grew at the same rate. But in the field, my artificial habitat seemed to attract a whole range of insects, including but not limited to 4 species of hoverfly and their associated parasites, but not the pine hoverfly. So I found it worked really well. It supported hundreds of individuals across a range of species, but just not for this particular species, which was frustrating at the time, but there it is. Now, these containers filled with decaying organic matter could mimic stump rot holes if they were filled with sawdust, as what I was trying to reproduce in the Caledonian pine situation. So, but then in bark lined tree holes, like in these images, filled with leaf litter. They could mimic more of these bark lined tree holes, which is what I was also trying to mimic. And then perhaps more like a ditch or a mini bog if filled with grass clippings. Um, and why do we want to encourage people to create these in their gardens? Well, it's because in many gardens in urban and rural spaces, this habitat is missing, largely because we've lost our mature trees, the veteran trees, and woodlands, and we tend to get rid of anything that's stagnant or decaying, fearing disease or fungus. We tend to drain the ground. Um, and I didn't invent this idea. The idea of creating like an artificial rot holes, hanging a bottle in a tree, for example, individuals were trying. As far as I know, nobody really tested it. Certainly not at the scale I was attempting with the Lagoons project. All it is a container with decaying vegetation and rainwater. But what I did come up with was the name for this as a wildlife gardening tool. And it's this name which I think has caused the reach and probably the success of the project. So I needed a name for what is essentially a rotting mini bog of eternal stench. And I'm not kidding about the stench, depending on what you fill it with. So if you don't use gloves, and I regularly don't when I'm messing around inside our garden, Hoverfly Lagoons, the smell from the bog-like lagoon can remain on your hands for some time, even after washing several times. Um, the sawdust-filled lagoons actually smell quite nice to me, but maybe this is a bit subjective. Um, but it needed to be, the name needed to be something positive that people might not at first be completely repelled by, never mind want to recreate in their gardens. And Hoverfly Lagoon made me imagine kind of a natural pool, perhaps where a hoverfly is bathing, perhaps sipping a cocktail, and this is, I asked ChatGPT 4 to recreate this image that I had in my head, and this is what it came up with. And of course, this is the wrong life stage. So I asked ChatGPT 4 if it could recreate this image with the correct life stage, the larval stage. And many, many iterations later, this is the best it could do. So clearly we've got some work to do with spreading the word. The most up-to-date ChatGPT AI has no idea what a hoverfly larva looks like, and wasn't very good at following my instructions. But this was deemed the cutest of all the iterations by my daughter, who is 9. The most appealing, perhaps, again, maybe it's subjective, but this is what citizen science needs to be: something appealing, something attractive that people will at least initially be keen to try out. People tend to like cute things, even bumblebees, they might as well be small mammals. And it's a hard sell counting maggots in a rot box, essentially. But there are a number of advantages to this citizen science project. First, it's very low cost. It costs pretty much nothing. We wanted people to use milk bottles to create the lagoons. The reason why is because pretty much everyone has a milk bottle that they're going to chuck in their recycling available to them to do it. This means that the shape and the size was standardized for what we wanted to do with the replication. But really, anybody, you can use anything as a container as long as it's a similar size, which is what we've moved on to now. And then everything else is stuff that you can collect in your garden, other than the pupation tray. So the pupation tray is there to sort of capture the larvae as they exit the water, because they want to pupate somewhere dry. And it really is just a tray. It can be anything as long as there's holes in the bottom. You can drill holes in the bottom, and then it can be kind of anything that you sit your lagoon in. So really zero cost. The methods are super easy to follow, and it's a relatively easy group to identify. I mean, you can, with the larval stage, there have been incidences where people don't know if it's a hoverfly, but generally it's very easy to, well, you can certainly tell from a photo. And I try to provide content to help people recognize a hoverfly versus a different kind of fly. It's for all outdoor spaces as well. As long as there's a space outdoors, you can have a hoverfly lagoon. It's also for all ages. And there's nothing else quite like it. It's novel. I wrote a piece for the IUCN report to the European Commission in 2022. And this report summarized a lack of awareness. And Hoverfly Lagoons, the project, was really the only example of conservation efforts in this way, reaching out to the public, involving the public in conservation efforts in Europe. So it's suitable for all ages. Most children love it. To be fair, most of these images are actually my children, but I have taken the project to schools and they do love it. The teachers, not quite as much as the kids, but they're usually talked over, brought over to the loving the maggot side, I mean. Easy for them to understand, the children, particularly because of the life stages. They're aware of butterflies, thanks to the very hungry caterpillar. So I refer to that a lot when I'm talking about life stages, life cycle of the hoverfly. And it's also an opportunity to talk about all kinds of things science related with children, the ecology and biology, introducing them to these things. But simply counting holds their attention when they're very young. Certainly with my children, when they were young, they'd be distracted by all sorts of things. I'd be desperately getting them to focus on whatever I was trying to show them, and me, a woodlouse. But they do, with the counting, that actually does bring back their attention, and generally they just sort of love taking part. Importantly, they notice things like the woodlouse. They observe, and working with them, it forces you to also notice things that we otherwise kind of take for granted, as we tend to take for granted the older we get. And I think this is another huge advantage of the Hoverfly Lagoons project, because it gets people out observing wildlife. And by doing this project, you really get an insight into a little known, little seen world. Really, it's a world of life in these small water bodies once you really start looking, particularly when you look really closely. And simply by observing and spending some time wandering around this world, you'll likely see something interesting or even incredible. So this is, I find making them with glass containers, they're not ideal for lagoons because they can crack. Certainly over winter, I wouldn't advise it, but during the spring and summer, it's great fun having a glass lagoon, a glass vase as a lagoon, because you can see the action and activity happening inside, and it can get really exciting at night as well. I've done some some footage at night of the activity happening inside lagoons. And these are eggs, and then the pupa and larvae in this slide, just to show you what they both look like. But you can notice incredible things. And there was a time where I was reviewing my lagoons. I was going over to count the larvae inside, and this Helophilus pendulus, otherwise the common name is the tiger hoverfly because of the stripes. And I took these series of images as it lay eggs or oviposited eggs on the underside of an ivy leaf. And this is a very common occurrence in my garden. There's lots of ivy, and my lagoons tend to be positioned under the ivy as shelter. And Helophilus pendulus likes to lay them in very beautiful, neat batches like this. And she will lay kind of up to 200 under a leaf, and the larvae hatch and drop into the lagoon that's underneath this overhanging. This is a fascinating insight into their ecology. When I first noticed this, I was beyond excited to see this myself. And I just think you can observe these things. And what you might think they do the same thing, like this is a similar niche, that surely the hoverflies are all behaving similarly within this niche that are semi-aquatic. But this behavior is unique to this group, the Helophilus group. This is what you might see more normally because it's not so noticeable, I guess, on the surface of the lagoon, just a complete mishmash, a great pile of eggs. And this is what Aristales do. So this is Aristales pertinax. I also caught her ovipositing, and I caught her before she'd finished. And you can actually see some eggs in the lid of the tube here where I'd caught her, because I couldn't quite see which species she was. I needed to know which species. Yeah, they dump their eggs just in this great mass, not neat and ordered like Helophilus. So seeing the adults in this situation is really, this is Myothropa florea, which is a huge, very beautiful hoverfly, maybe in contrast to the larval stage, right, which people are not quite. You can, I mean, it's subjective again. That's the bit whether you say this is beautiful or not. But the lagoons, spending time with them, it allows you to see these different species, the differences, as well as many other insects and more broadly invertebrates that will visit these small water bodies, either using them as a breeding habitat or as a water source. And small water bodies are really important, and even something this small can attract a range of animals. And here, this isn't Myothropa florea feeding in this lagoon. These are wasps, and they're just there having a drink. So as far as the idea goes, we thought it was a good one for the Buzz Club. And these are methods I created, and actually they haven't changed since I first created them. I think it was around 2016. And they were designed in a way that hopefully was really easy to follow, that was appealing to look at. Although, I mean, I can see now things that I would want to change if I had some time to make them look just a bit more professional. I know Bug Life have replicated these a little bit for their um, for for their purposes, and they do look a bit more professional. But we do at Buzz Club now have a working group, which is a brilliant idea, new to this year, who give comments and suggestions to help us design our methods and make sure everything is clear. And they give some really, really useful feedback. And the feedback for this year, which included much of the same methods, was all positive. So already mentioned is this value of having many people to do it. And this is true of the Hoverfly Lagoons project as well. We couldn't achieve what we're trying to do without the number of people, the scale. Beth and Dave both mentioned this. So the protocol needs replication. And also, I hope in the future it can be used as a way to detect changes in across locations in the UK, changes in life stages. Um, so we might be able to see changes in the things of in the timing of life stages in relation to changes in climate, and I think it could be a really useful tool for that. And hopeful in the future, we just need people to do it and continue to do it over years. And this is the spread of the number of volunteers that we're doing it. I think this was 2017, so we had some in Scotland, but most of them were in England. There are almost limitless questions you could ask with hoverfly lagoons, such as looking at the size. Well, first of all, we looked at whether it worked at all. We wanted to know if this actually did work, if people could create them, if hoverflies came, and of course they did. Is size important? Is position important? And so on. Some of these questions my research students at the university explored rather than citizens on the project. So the mosquito question, how do we limit mosquitoes? Essentially, how do we prevent this from being a mosquito farm rather than habitat for hoverflies? And whether they had a positive effect on strawberry pollination. Because these questions are a bit more complicated. In terms of what we're asking people to do, it needs to be simple and clear for them to follow, not a chore, not a task either. And my students' findings very much support the project, providing more information and context, as well as helping with the appeal to create lagoons in the first place and helping with outreach. In terms of the findings, just a summary of the findings here, because there's not any time to go into this in any detail, but 8 years on, and the Hoverfly Lagoons project has confirmed 7 species using artificial habitat. This is, there are potentially up to sort of 20-25 species that could use artificial tree holes. We've recorded 285 larvae in one lagoon. That's the maximum, I think. We've detected population booms in one of the species, Cerita pipiens. They boomed in 2018 and 2020 across locations in the UK, which is really cool to see. And we discovered the previously undescribed pupal stage of Ringia rostrata. That's this hoverfly here. And this is published now in a little known journal called Dictors Digest. This is the pupa here to compare with Myothropa, which is much, much bigger. So new to science. If you're interested to know more about Lagoon's findings, Kieran mentioned Entolive at the beginning. There is an Entolive talk I'll be giving in July where I will go into more depth with these findings. For more information and detail about some of the things that I'm talking about here, I've got a blog website as well. So do go there if you want to have a look at the species that we found and some of my blogs at hoverflylagoons.co.uk. This was designed to support the Buzz Club website to provide more detail. So possibly one of the major results or successes of the project is its reach. And this is why I've titled the talk, Spreading the Word. And I suspect this has a lot to do with the name. It was picked up by the media somewhat here in the Sunday Post and gardenersworld.com, and online is where probably it's done most of its traveling. Probably because it's quite a novel, unique, easy gardening wildlife tool. Searching the term online and on YouTube, people have made their own methods for hoverfly lagoons, and I love this. I love seeing what people have done in their own interpretation, and just the fact that they want to do this and spread the word and get people excited about it makes me very happy. We don't have, or we haven't had many Welsh volunteers, but last year I was contacted by an organization that offered to translate the methods to help change that, I guess. Or, you know, as long as people are making the habitat, ideally we'd want them to join the project so we can improve the data. But I thought this was great, very cool and exciting. Habitat managers have contacted me, and here on that blog website I mentioned is a story about a farmer in Norway who was very into making lagoons. In this case, it was actually boring holes in stumps rather than plastic containers or some other container, but it worked very well, and he saw it right through, collecting the pupae at the end and everything. It was very successful. I made a guidance document for forestry managers as well in this way, and really it is kind of an attempt to bolster biodiversity at these sites. The lagoons method actually made it all the way to Australia. In Tasmania, believe it or not, there is some program on, and they mention lagoons and talking about it. The professor who is involved in this research in Tasmania contacted me to ask me about the methods and details. And when you thought it surely couldn't travel further, yes, it can. It did. Asking for permission to use. Just when you thought it couldn't get any further than Tasmania, here it is in New Zealand. Yeah, they wanted to follow the methods. They wouldn't be included in the project, but I think this is a big part of the project generally, just that outreach. And it continues. Schools are increasingly interested. There's PhD students including them in their research, which is so cool. So I've been advising them a little bit in Scotland, particularly this year, and that inspired actually the Hoverfly Lagoons project with